today we're going to finish up the Chapter 8 lecture. Again, if you didn't watch the videos, the end of Chapter 7, the beginning of Chapter 8, 2 videos, I've just labeled them as Monday's virtual lecture. Make sure you watch those. So the Chapter 8, the carbohydrate catabolism or metabolism, cellular respiration, I got up until this slide. So everything in the beginning of the video was going through a lot about what are enzymes, what are some of the processes that are taking place when we're breaking things down, building things up. And then I want to finish right to here because carbohydrate catabolism, I like to draw out on the board. Now, what is catabolism? And it's the opposite of anabolism. Things down. So carbohydrate catabolism is we are breaking down carbohydrates specifically. Lots of organisms like to oxidize carbohydrates. That's losing electrons. Pretty much when you break a bond, you lose electrons. Something else is going to be right there to pick it up. But we're breaking down carbohydrates, releasing electrons. Glucose is the most common carbohydrate that is used. Does anyone remember what the chemical formula for glucose is? Yeah, I was going to say, I drew it out in class. Or wrote it out. C6H12O6 is glucose. And so we're going to be breaking down glucose and glucose catabolism. And we're going to look to see when glucose gets catabolized and broken down, what happens? Like, where does that energy from releasing electrons go? What picks it up? How can our cells make ATP, which is our more energy currency than just the energy that's stored in those bonds? Now, as glucose gets broken down, there are 2 processes that it can break it down with: cellular respiration and fermentation. Cellular respiration can happen aerobically or anaerobically. Fermentation, that specific fermentation process, only happens anaerobically. Now, I'm handing this out. This is for you just to write on. I'm not collecting it back, so I don't care what you write all over it. I'm going to draw stuff on the board. And if you're a person that likes to draw stuff and you didn't bring any other paper, you can use the back side of this worksheet. Just note, I still don't have a ton of board room for what I want to draw, but I'm going to draw this way because I'm going to use up every little space of my board up there that I can. And I like to get colorful too. But as we break down glucose, there's multiple steps that are happening. And so this table is just trying to help organize that information. You're welcome. Hopefully I had enough. Ooh. Here, have this. Maybe. Now, we're going to go through all of the steps that are up here on this picture, this diagram, but I'm going to refer back to the diagram. There's lots of steps. We're going to break it down step by step. Just note, at the very, very top is that glucose molecule. And depending on whether this glucose molecule goes onto this green area, that's your cellular respiration, or whether the glucose then is going to follow and do the orange top square, that's fermentation. Less steps, a little simpler. We're going to be following it through. So glucose, no matter what, is either going to be the starting process and it's going to get broken down and it's going to go the cellular respiration route, or glucose is going to get broken down and it's going to go the fermentation route. We're going to start doing the green side, the bigger side, and going through glycolysis, this first little square. It's going to go into the Krebs cycle. It's going to do the electron transport chain. That's cellular respiration. Versus fermentation is just glycolysis and then further broken down. It's shorter, so we're going to start with the longer part first. Now, there's a lot of stuff up on that image. Most of it I don't care about. Majority of it I don't care about. Because as I gave you that worksheet, mostly I want to know where did the different steps take place, what goes in, what comes out, and am I like anything else important, and how much energy do we get out of these different steps. So at the very, very top, we're taking the glucose molecule, the very, very top, And pretty much all we're doing as we go through it and breaking down glucose is we're following the 6 carbons as they get broken down. And so the 6 carbons are always drawn as 6 black little circles, and I'm going to do the same. Here's my 6 carbon molecule. There's my glucose. The word glycolysis, lysis means to break apart. If you lyse something, you break it apart. So glycolysis is breaking down glucose. That's just what the word means. We're breaking it down. Now, lots of steps before we get to our final end product. And to signify there's lots of steps that I don't really care about, there's a bunch of little arrows there. The end product we get out of the process of glycolysis, the final end product, is we get 2 3-carbon molecules. So all the carbons are still present, still drawing 6 circles. But they've been split into 2 3-carbon molecules called pyruvic acid. Some books call it pyruvate. It's the same thing. Just note if you see pyruvate, it's the same thing as pyruvic acid. It gets 2 names. I don't know why. But other than the starting material and our final ending material, there's other stuff that's happening in here. And it gets colorful up there, and I'm going to get colorful as well. I got lots of colors I'm going to use today. But looking at the arrow-wise and that picture, near the very top, what has to go into the equation? ATP. And how many ATP at the very top are going in? 2. They use kind of like orange boxes, so I'll use orange. Why do we need ATP to go in? Need energy to start a reaction. Reactions don't just spontaneously begin. We need some of that activation energy that's going to get this reaction going. Now, our purpose is to break down glucose and get energy, and right now at this point all we've done is put in energy. But we've got to put a little bit of energy in, just a little bit, just to get this thing going. Now, looking at ATP near the bottom, how many ATP do we get out that you see coming out arrow? Plus another 2, we get 4. We've got 2 ATP coming out here, and we also have another 2 ATP coming out there. So at least we're ahead when it comes to ATP. What's something else we see coming out of the equations? Water. How many waters? 2. Like them do. What else? 2 NADHs. Why do I care about NADH? Yes, it's an electron carrier. It's what's going to grab electrons. Again, as you break bonds, you do release electrons, and there needs to be something right there to pick them up. Well, NAD was right there to pick them up, and it became NADH. It picks up a couple of electrons, and it picks up a hydrogen that goes along with it to make it stable. So now we've made some things that are some electron carriers. Now, this whole process in the cell happens in the cytoplasm. And we've talked about 2 kinds of cells, eukaryotic cells and prokaryotic cells. Do both cells have cytoplasm? Yes. And I'm like, so it doesn't matter whether it's a prokaryotic cell or whether it's a eukaryotic cell, this process is still going to happen in the cytoplasm. Now, making sure you've got everything filled in on your worksheet. So where does this all happen for glycolysis? Cytoplasm. For what goes in, the 2 main things I just want you to know that goes in is the starting material, so that glucose molecule. So 1 glucose molecule and 2 ATP, because it won't start without that. For what comes out, it's everything that I drew an arrow coming out. So we've got 4 ATP came out, 2 NADHs, 2 H2Os, and 2 pyruvic acids. A lot of stuff coming out. If a glucose molecule went in and a couple ATPs, and we get a lot of stuff coming out of this reaction. For the box that asks energy produced, did we produce some energy? Did we produce some ATP? We did. We got some ATP out. How many ATP have we gained, net gained, out of this process? Just 2. So we've gained a total of 2 ATP. We put 2 in, we got 4 out, we're ahead. We've netted 2 ATP. For my anything else important, one of the most important parts of glycolysis, that's this box in the middle. Glycolysis is part of cellular respiration. It is also part of fermentation. So breaking down glucose and making pyruvic acid, depending on where those pyruvic acid are, they're either going to go into the cellular respiration, that whole green area of the box, or they're going to go over into that orange area of the box. Glycolysis is the first step for either process. Which then also answers kind of that last question, or I didn't have another one. I used to ask, is it part of fermentation or is it part of cellular respiration? It's part of everything. But we're going to start with the green, so we're going to follow them through. And those pyruvic acid, as it goes into the cellular respiration state, so I kind of got rid of the fermentation step, it's going to further get broken down. It's going to become acetyl-CoA. It's going to go into the Krebs cycle, and then it's going to go through the whole electron transport chain. So we're going to follow it through those next 3 steps. Here's my 1, 2, 3. So pyruvate is the same thing as pyruvic acid. And as I draw this out, again, I'm going to use all of the rest of this board. I'm still going to run out of room. Although there are 2 pyruvic acids here, I'm just going to pick one and follow it through. Otherwise, my board would get even more chaotic. Just note, whatever I draw that's happening to this pyruvic acid is also happening to the other one. So when we start to count up numbers, whatever's happening here is also happening to this one. But I'm just going to draw out one. So at the very top on the left-hand side there's pyruvate, same thing as pyruvic acid. And it undergoes, I'll follow this top one. It's got some different things that are happening. But the final outcome is a 2-carbon molecule called acetyl-CoA. Now, not as much is happening in going from pyruvic acid to acetyl-CoA. But what's something we get out? That we see arrows. We get some CO2 out. Now, this explains why we just went from a 3-carbon molecule to a 2-carbon molecule. Well, where'd that carbon go? It didn't magically disappear. It's what gets breathed out as part of your CO2. And so I need another color. We'll use red. We do lose one of the carbons by releasing carbon dioxide. What else do we get out? It's another arrow coming out. NADH. How many are we getting out on that picture? One. So 1 pyruvic acid makes 1 NADH. Now, I don't care about the CoA going in. That's part of what makes that acetylcholine, or that coenzyme A. But where it takes place. So depending on the organism, I shouldn't even say it. It doesn't matter whether it's a prokaryotic organism or a eukaryotic organism. This also happens in the cytoplasm. For the what goes in, there's not a lot going in. There's only one thing that goes in. What is it? The pyruvic acid or the pyruvate? 2 pyruvates specifically. Again, we're following the breakdown of one glucose molecule, and even though I'm only going to follow one of the pyruvates or pyruvic acids through, both are going in. Both are getting broken down. So what goes in is 2 pyruvic acids are each getting broken down. For the what comes out, what's one thing that comes out? There's 3 things. 2 what? 2 carbon dioxides, so 2 CO2s, one here and then one that's also happening here that I'm not going to draw. What else comes out? 2 NADHs, one here and one in this other one I'm just not going to draw out. And what else? Yeah, the acetyl coenzyme A, acetyl-CoA. 2 of them. Again, we're breaking down one glucose. We're looking at what are we all getting out of that one glucose molecule. I'm just only following one pyruvic acid through. But whatever's happening here is also happening here. It just would look super crazy, especially as I continue to the next step. Any energy? No. No ATP whatsoever. None, nada, nothing, however you want to write that in there. But anything else important? I kind of put other information. Is acetyl-CoA, once you've made acetyl-CoA, it will go directly to the Krebs cycle. So acetyl-CoA is not just going to float around hanging out in the cytoplasm. It wants to go to the next step. It will directly go to the Krebs cycle. Here's my Krebs cycle. So here's my acetyl coenzyme A, and it's going to go into the cycle. On the picture, it has it drawn going in at the very top. So I'm also going to have it drawn going in at the very top. And it's a 2-carbon molecule that goes in, the acetyl-CoA. But there's another 4-carbon molecule that hangs out in this. I don't care that you know the name. It's the oxaloacetic acid or oxaloacetate. But that's a 4-carbon molecule. It's got the 4 little black circles on there. And that 4-carbon molecule will combine with this 2-carbon molecule, and now how many carbons do you have? You're back to a 6-carbon molecule. It is not structurally the same as glucose, but it does have 6 carbons. This 6 molecule is called citric acid, which is why sometimes the Krebs cycle is also called the citric acid cycle. So lots of things are happening. I do not care about all the intermediate steps, and if you're like, wow, but that all looks really interesting, you can take a cell biology class at UWL. That all it is everything about the cell, and you can learn all the intermediate steps. I mostly care about, as it goes through all of these steps, what are we getting out? What do we get out of breaking down this new citric acid molecule that we just made? What is one thing that you see coming out? So lots of things coming out. What's one? NADH. How many? 3 of them, because there's one right there, and then one way up at the top up there. I'm not going to draw them all separately. I'm just going to say 3 NADHs are coming out. What else comes out? What? FADH2, how many? Just one, and I think I ran out of colors. Coming out right there. Why do we care about FADH2? It's also an electron carrier. What else comes out? ATP. How many? Just one. One little ATP down there. And there's something else that's coming out, and it always comes out when NADHs come out. CO2, or I guess not all of them. How many CO2s come out? 2 of them. One comes out when it comes out with that NADH, one comes out when it comes out with that NADH. Now, we need 2 carbon dioxides to come out. Again, we now have a 6-carbon molecule. That 2-carbon molecule plus that 4-carbon molecule gave us a 6-carbon molecule, but it's called a cycle. This will keep going, and that 6-carbon molecule will eventually go back and become that 4-carbon. As you go from 6 to 4, those 2 carbons, again, don't magically disappear. They get lost as some of that CO2. Now, for filling in your table, where does this happen? And this will make a difference whether it's a prokaryotic cell or a eukaryotic cell. Eukaryotic cell, for everyone that just recently took advanced A&P or maybe is suffering through it right now. I don't know if you've gotten to this part yet. Where does it happen in a eukaryotic cell? Yeah, and specifically which? It does happen in the mitochondria. It happens in the fluid or the matrix of the mitochondria. But does a prokaryotic cell have a mitochondria? No. But it still can go through the Krebs cycle. In a prokaryotic cell, it's still going to happen in the cytoplasm. Now, for the what goes in, the only thing I really care about that goes in is 2-acetyl-CoA. The one that I drew and the other one that came from that other pyruvic acid. So 2-acetyl-CoAs are both going into the Krebs cycle. I know a couple water go in as well, but I don't really care. It helps break down a final couple bonds, but the big thing that goes in is those 2-acetyl-CoAs that we get out of our glucose molecule. What comes out becomes a lot more important with the Krebs cycle because we get a lot out, especially number-wise. What's one thing we get out? We've got 4 carbon dioxides. 2 here and 2 in the other Krebs cycle I didn't draw. Now, how many carbon dioxides total have I gotten out of everything so far? 6. That is then taking care of all 6 carbons out of my original glucose. So we're not going to lose any more carbon dioxide after this. So all 6 carbons at this point have been lost as CO2. What else do we get out? Total number-wise. Those are too quiet. What else do we get out? 2 ATP. 1 here, 1 in the other Krebs cycle I didn't draw, but it's still happening. What else? 6 NADHs. 3 here, 3 in the other one that I'm not drawing. Again, if I tried to draw both, this board would look crazy. And there's one more thing. 2 FADH2s. For the question of energy produced, did we get some energy out of here? Yeah, not a lot. We got 2 ATPs out of the breakdown of glucose. 1 from one Krebs cycle, 1 from the other Krebs cycle. We get 2 ATP out. Still not a lot. For anything else important, I put it on here. It's wordy. I didn't know what it was. The big thing that we're really getting out of that I say is the most important is we now have a lot of energy in our electron carriers. So that's that first statement. It transfers most of the energy as we're finally breaking everything else down. It transfers most of that energy into making NADH and FADH2. And altogether, we've got 6 NADHs and 2 FADH2s from this step. It's a lot of electron carriers. It's a lot of energy in electrons. And we also get several precursor molecules. Each one of these steps breaks down into something else that could potentially be used somewhere else. Our last step, and I did not leave myself a lot of room, is the electron transport chain. I really want to draw it over here. I'll try to draw it kind of small. And we'll go purple. I'll draw another one. And I'm not artistic, just FYI. Not an art person. Here's my phospholipids. And as books usually have proteins as big purple blobs, there's some proteins. I'm going to skip ahead and come back. See, like, this is what I was trying to draw. You got some big proteins, big blue blobs, big purple blobs, but you also have this one very specific protein called ATP synthase. Again, it's an enzyme. It likes to end in ACE. And enzymes usually tell you what molecule they're working with or what do they do. Based on the name of this enzyme, ATP synthase. And guess what the job of this enzyme is? Makes ATP. Synthesizes or makes ATP. So the job of this enzyme really is to be making lots of ATP. Now, it's called the electron transport chain. And so we have a lot of electron carriers that are all going to make their way to the electron transport chain. Whether it's the FADH2s, whether it's the NADHs. They're carrying electrons that they picked up from the breakdown of that glucose. And they're bringing those electrons and the energy that's in them. And it's also bringing the hydrogens that were attached to them as well. Those electron carriers are going to use the energy in those electrons to pump. Here's my NADH, FADH2. It's going to use the energy that's in the electrons in there to pump hydrogens across the membrane. So it's using the energy of the electrons to pump hydrogens across the membranes. So now we have an area where you have lots and lots of hydrogen ions. Do things like to be crowded? No. So these hydrogen ions, across, they're all very concentrated over here. They would like to sneak back to the other side where it's not so hydrogen concentrated. ATP synthase has a hole in it. That hydrogen ion can sneak back through the ATP synthase protein, it's an enzyme, and when it goes through there, the top of this spins like a turbine. So as these super concentrated hydrogen ions are all over on this one side, they want to sneak back. They're going to go through this enzyme. They're going to spin the top of that enzyme, and the spinning of the turbine is going to generate the energy to take ADP with another phosphate and make it an ATP, adenosine triphosphate. So we broke down glucose. We had a ton of electron carriers all picking up the energy of those electrons from breaking bonds. It also picked up some hydrogen ions with it to make it stable. It brought all of those electron carriers, little taxi cabs, bring those electrons and hydrogen ions, take the energy of the electrons, pump hydrogens across, purposely to create a gradient, which just means you have lots of hydrogens on one side and very few on the other side, which forces those hydrogens to want to sneak through that one ATP synthase enzyme. Spins the turbine, generates ATP. Now, at some point, the electrons, they're taking their energy and they are pumping the hydrogens across. At some point, those electrons can't just float around by themselves. They do have to bind up to something, because electrons, if they're by themselves, become volatile. They usually bind up and will form water. They usually will bind up with a hydrogen, with an oxygen molecule, if oxygen is present, and form water. But I want to go back to where does it happen? And what do we get out and what goes in? So again, this makes a difference whether it's a prokaryotic cell or it's a eukaryotic cell. If this is a eukaryotic cell, this phospholipids is happening on the inner membrane of the mitochondria. So inside the mitochondria, it's got all of that surface area on that inner membrane. So in a eukaryotic cell, the inner membrane of mitochondria. But as we've already established, the prokaryotic cell does not have a mitochondria, so it can't happen on the inner membrane of a mitochondria. Do you remember from one of our first lectures where ATP is made in a prokaryotic cell? Out on the cell membrane. The outside part of the cell is still a phospholipid membrane. So in a prokaryotic cell, this is just going to happen out on that plasma membrane, the cell membrane. It's the electron transport chain. So what goes in are all of our electron carriers. Add up all of the electron carriers that we've generated from previous steps and how many go into the electron transport chain. How many NADHs do we get if you add them up from all the steps? 10 NADHs, because we had 2 from the glycolysis, we had 2 from acetyl-CoA, and we had 6 from the Krebs cycle. How many FADH2s do we get? Total? 2. Just the 2 that came from the 2 Krebs cycles. It's 2 FADH2s. So what goes into the electron transport chain are all the electron transporters. It's the 10 NADHs and 2 FADH2s. What comes out? ATP. And a lot of it. So this also goes down to its energy produced. Depending on the organism, it can generate anywhere between 32 and 36 ATP. It's a variable number. On average, it's 34. But there are some organisms that they kind of have leaky membranes and doesn't make as much. Because sometimes hydrogens can sneak back without going through that enzyme, which means you're going to have a lower number. Some are more efficient at pumping them right back through, and they get a higher number of 36. Average is 34. So we also get water as well if oxygen is present. So again, those final electrons have to find some type of final electron acceptor. As long as there's oxygen present, we do get some water out as well. It's not as important, but most of these processes happen aerobically. For the energy, yes, the 32 to 36 ATP, we get lots of ATP out of the step. For anything else important, the electrons from the electron transport chain have to find a final electron carrier. Again, if there's oxygen present, that's when it makes water. But we'll talk about things when oxygen isn't present, what can happen. It takes those electrons and their energy to pump hydrogen ions across the membrane. That ATP synthase enzyme makes ATP from the hydrogen ion gradient, meaning you've got lots of hydrogens on one side, not a lot on the other side. They sneak through and they make lots of ATP. Lots of little things that happen in there. I think one of the book pictures, I think has this as that picture. You know, like that's a horrible picture. That looks way worse. I think this was from an old textbook. I liked this better because I can somewhat try to draw it in my little tiny room that I had left. Now, for that final electron transport, again, if this is happening aerobically, meaning there is oxygen present, oxygen serves as the final electron carrier. It's when we get water out of it. And if oxygen is present, we will have a higher yield of ATP. Eukaryotic cells, bacteria, can do some of these steps anaerobically. We can't. Like, we need oxygen to survive. But there are some bacteria that can survive anaerobically, and they can do this. It is not as efficient. Meaning anaerobically, as some anaerobic bacteria can do, again, we can't, but some anaerobic bacteria can do this. An inorganic molecule, that's not oxygen, because inorganic in theory just means not carbon-based, but an inorganic molecule that's not oxygen serves as some type of final electron carrier. So maybe it's a hydrogen, maybe it's a nitrogen. But some other inorganic molecule that's not oxygen will serve as the final electron, the electron carrier, if it's happening anaerobically. We are going to flip gears and talk at fermentation. And since I was looking at final electron carriers, I threw in the final electron carrier in fermentation is an organic carbon-based molecule. So kind of just comparing is as you're breaking things and releasing electrons, at some point they have to find a final resting place. And depending on the organism and how it does these different steps depends on what the final resting place is. So we're going to flip to fermentation. I'm going to erase a bunch of this, but not all of it. Why am I leaving glycolysis up there? Because what? It's the start of fermentation. If I can go back. Just showing the fermentation, the right-hand side, glycolysis is still the very first part of fermentation. Now, for glycolysis to take place and NADHs to get made, an empty electron carrier to pick up the electrons. My little thing, before we can make any NADHs, there does have to be an empty. It's like the empty taxicab that now is picking up electrons and hydrogens. So cells do require a constant source of NAD+ for glycolysis to happen. So this can't happen. We can't make NADHs if we don't have the empty taxicabs that are just ready right there. Fermentation, the process of fermentation, is going to provide these empty taxicabs, those NAD+, those empty electron carriers. The whole process of fermentation is a partial oxidation. It's just a partial breakdown of the glucose. It does not have as many steps to fully break down every single carbon molecule. It just partially breaks it down. And it's going to use an organic molecule as a final electron acceptor, which I already stated on that other slide. So it's going to take the pyruvic acid that it gets from the breaking down of glucose, and it's going to turn it into something else. The biggest thing is it depends on what organism it is and depends on what it makes. In the body, that you guys study in A&P, in the body, it generally makes lactic acid. But in our class, that is not the only option. In the human body, it is. But in our class, depends which microbe is taking pyruvic acid, depends on what it can make, and it can make a lot of different things that we use on an everyday basis. Now, our net energy gain, I'm going to go back and forth with this. This is showing an example that pyruvic acid, just two pathways among multiple number of pathways. One pyruvic acid, there's another one too. Depending on the organism, the human body will make lactic acid. And it's a carbon-based molecule that's going to pick up that final electron acceptor. It's an organic molecule. But it gives us that NAD+. Other molecules, other organisms out there can take pyruvic acid. It will still give us NAD+, so that I can go back into glycolysis. Also make some CO2, but can make ethanol, alcohol. Now, the CO2, again, that's part of it's an additional breakdown of that pyruvic acid. But it's not the only two pathways. It just is two examples of what can happen with pyruvic acid. However, in the breaking down of pyruvic acid to either make lactic acid or to make ethanol, is any ATP made in these steps? No. That doesn't mean the process of fermentation doesn't make ATP, because the very first step of fermentation is glycolysis, and do we make ATP out of glycolysis? We get 2 ATP, a net of 2, from glycolysis. So the whole process of fermentation only has a net of 2 ATP, and that's just from that first glycolysis step. Now, this is showing different examples of who can do what with pyruvic acid. So this is showing fermentation. Glucose is broken down into pyruvic acid. Propionibacterium can take pyruvic acid. They can break it further down, and they can make what's known as propionic acid and CO2, kind of like the alcohol releases CO2, and it gives that sharp, dry flavor to Swiss cheese. If you ever wondered why there's holes in your Swiss cheese, that's the CO2. There was little gas bubbles or gas pockets in that cheese. And the flavor of this particular acid gives Swiss cheese its flavor. So this particular organism, very specific bacteria, makes your Swiss cheese Swiss cheese. Aspergillus, Lactobacillus, and Streptococcus species can take pyruvic acid and make lactic acid, just like our cells can make lactic acid. However, different organisms and where they are used and what they are used with, it gives it the sharp flavor used in cheddar cheeses, yogurts, and soy sauce. Saccharomyces is a yeast. So that organism can take pyruvic acid, make it into CO2 and ethanol. That's your alcohol that's drinkable. Clostridium, not C. diff though, but there are other Clostridium species that can take pyruvic acid and make it into acetone, an isopropyl alcohol. So different organisms can take pyruvic acid and make different end products. Again, it's just taking that pyruvic acid, further breaking it down, and depending which organism, what breaks it down, and what the final electron organic molecule is, depends on what we can use it for. So we use bacteria in industry for a lot of different things. Now, away from catabolic pathways, away from fermentation, away from cellular respiration, flipping gears, which I don't have a lot of slides on. Most of what we talk about in this PowerPoint is breaking stuff down, releasing energy. An anabolic pathway, it's the opposite of a catabolic pathway. It is building something up from smaller molecules. It's taking a whole bunch of small things, putting them together to make one larger molecule. But if you build things, it requires energy to go in. I can't just take a bunch of bricks and be like, okay, assemble yourself. It takes a ton of energy to actually build a building. Where the energy comes from is from a catabolic pathway. As you break things down and release energy, electrons, they can go through that whole process. They can make ATP, and that ATP can then be used to build other things. So when our body wants to assemble a bunch of amino acids into one large protein, or it wants to make some other structural material, it takes the energy from breaking things down. This is why we eat. We eat, we digest, it's breaking things down, it releases the energy from the food, and then it gets stored until we then need that energy to do something. Now, anabolic pathways are the reverse of a catabolic pathway. Again, where something gets broken down, it can then get reassembled. So this is showing two small molecules, molecule A and molecule B. Two individual molecules can get put together into one larger molecule called AB. But it can also go the other way, and it can get broken down into its individual basic parts. Just note, different enzymes have to make that happen. One enzyme is used to build things up. Another enzyme is used to break things down. It's not the same enzyme. ATP synthase cannot break down ATP. Its job is only to assemble ATP. So the reactions can always go opposite. Catabolic and anabolic, you break things down, you build things up, but you need different enzymes to do it. Now, lots of words on the slide. I'm sorry, it's like, one of those things on, you shouldn't have too many words on a slide. It's called integration and regulation of metabolic function. Or I like to call it how our cells like to be lazy, or efficient. Our cells don't want to put any more energy into anything that they don't absolutely have to. So these are all different ways on how our cells try to be efficient, lazy. So one, cells are only going to synthesize or make enzymes if the substrate is available. If we're trying to break down a particular substance, a sugar, whatever, even if it's glucose, if glucose isn't present, why make the enzyme to break down glucose if glucose isn't around? So cells aren't going to make enzymes unless the substrate's there. Cells are going to catabolize or break down the more energy efficient choice if more than one is available. If glucose is present here, if there are other sugars present there, the cell will break down the most energy efficient one. Cells are going to synthesize or make metabolites they need. So they're going to make the different molecules they need, and then they're going to cease the synthesis if the metabolite is present or available. Okay, we're going to make what we need to make, and then as soon as it's there, we're going to stop making it. It's wordy. Let's make what we need, and then once we've made enough of it, let's stop. And a lot of that goes back to once we've made whatever our cell needs, a lot of times that final product that we need is feedback inhibition, meaning that final product stops our cells from making any more of it. Why make a whole bunch of one type of sugar and just keep making it and keep making it? At some point, it's just wasteful. It's wasteful energy, space. Once we made enough of something, that tells our cells to go back and stop making it. Again, how cells like to be lazy, efficient. And this last slide, I think it's my last slide. I think I had a couple review questions maybe. It's just showing everything that's a purple arrow is a catabolic pathway. Everything that's a green arrow is an anabolic pathway. And as we go through the glycolysis stage, the Krebs cycle, we can see there are multiple little steps. I didn't draw all of them out, but lots of little steps that we can get made from breaking things down, but we can go in reverse and make it an anabolic pathway. We can get some of these different molecules from the breakdown of other things, and they can, again, get used to make something else. Our cells are constantly breaking things down into different molecules and then using those basic building blocks to build something else. It's just like if I was going to destroy this building and break it down into all the bricks, I could take all those bricks and go somewhere else and I could build another building. And so we're constantly breaking things down, making those smaller building blocks, and then building them up to make whatever it is that our cell needs to make. And that's where we're ending today.